Hi everyone, I'm Eddie,
I'm Ashwin.
And I'm Raj.
and welcome to Blood Cancer Talks. Of course, this is a podcast dedicated to hematologic malignancies, where we usually bring content experts who live and breathe a particular disease and focus on the latest advances in biology and clinical management. But today we have a slightly different format for you. Some of you may listen to a podcast called The Rest is Politics. Certainly I do, very different topic. And don't worry, we're not turning into a politics podcast. But we are recording an emergency podcast. So when something happens in the world of politics, they will drop everything and record an emergency podcast. Now, we, of course, have day jobs, so we can't quite drop everything. But we're coming to you very fresh off the back of the Food and Drug Administration, the FDA's Oncologic Drug Advisory Committee meetings, which were held last week. They held two hematologic ODACs and two solid tumor ones we won't be talking about. First, on the AQUILA trial in smoldering multiple myeloma, and second, the STARGLO trial in second-line or relapse-refractory diffuse large B-cell lymphoma. And because we thought that both of these discussions were so relevant, we thought we'd put together a little emergency podcast. So it's a bit of an experiment on our part. We're not sure how it will go, so please feel free to reach out to any of us and let us know what you think. And here goes. We hope you like this trial of a slightly different format, but don't worry, we've got plenty more expert episodes coming to you soon. So over to you, Raj.
All right. So let's see how it goes today. So I will start with the AQUILA trial. Just to give a background on the AQUILA trial, we will discuss the top line results first. As many of you know, AQUILA trial was a phase 3 randomized control trial comparing single agent daratumumab for 3 years versus active surveillance for quote unquote high risk smoldering myeloma. And we'll go into that as to what that means. It met its primary endpoint of PFS. The 5-year PFS in the daratumumab arm was 63% versus approximately 41% in the control arm. And I will recommend listeners to tune into our episode 55 on ASH 2024 updates with Dr. Popat, where we had discussed the top line results of AQUILA after ASH. However, as we all know, the treatment of high-risk smoldering myeloma is an extremely controversial and contentious topic, and a PFS benefit is usually not enough to convince the myeloma community to treat an asymptomatic population. As we have already seen with a slew of prior trials like the E3A06 trial, which compared Len versus observation, which also showed a PFS benefit, it did not really change the practice broadly. Yes, some people are using it, but overall, most are still undergoing observation. So the question that the FDA had was whether the benefit risk profile was favorable for daratumumab in high risk smoldering myeloma. So what I will do is we'll go over the key concerns raised by the FDA reviewers for approving daratumumab in high risk smoldering multiple myeloma, and then we can also go over some of the counterpoints that the sponsor made or some of the other experts in the panel made and have a discussion regarding the overall data.
So Raj, you said high risk, quote unquote. Why did you say high risk, quote unquote? What did you make of their definition of high risk in the AQUILA trial?
Yeah, so that's a nice segue to talk about the high risk populations, which was one of the concerns raised by the FDA reviewers. As we all know, the current definition for high risk smoldering myeloma that we are using is the Mayo 2018 criteria and then the IMWG criteria, which was kind of a validation of the Mayo 2018 criteria, and it just added one more factor, the FISH cytogenetics, to it. So both of them were published after 2018, and this trial started accruing in 2017. So when this trial was designed, those high-risk smoldering myeloma criteria were not there. And the criteria before that were the Mayo 2008 criteria and a Spanish criteria. However, one of the problems was that in the inclusion criteria for this trial was, I would say, a mishmash of all of the different criteria, but did not really align to one particular criteria. And it included, several things, for example, immunoparesis, IgA subtype. A lot of these factors we don't typically use for definition of high-risk smoldering myeloma. So that was one of the concerns raised. And typically nowadays, the consensus is that no matter which risk stratification we use, if we are able to identify a group which has about a 50% risk of progression from smoldering myeloma to myeloma over 2 years, that should be good enough. That's considered a high risk smoldering myeloma group. But if you look at the observation arm of the AQUILA trial, the risk of progression at 5 years was roughly about 50 to 60%. So not at 2 years. So that was one of the things that was a point made by the FDA reviewers, that definitely there were a lot of patients included in this trial that, may have been much lower risk than what we would now consider suitable for intervention. And only about 41% of the population were actually the Mayo 2018 high risk. So, that's why I said quote unquote, because this was not a trial purely of high risk smoldering myeloma population the way we would define it today.
So I have one comment and one question. My comment is that I think an important flip side to that is that almost half of these patients in 5 years in the active surveillance arm definitely didn't need treatment because they didn't even progress by SLIM criteria. So there's if you're going to start treating people, there's going to be a group of people that you might overtreat. But my question, Raj, is if they had used more modern definition for high risk, how do you think that would have affected the results?
So I think if they had used a more modern definition like the Mayo 2018 criteria, this trial would have been, definitely would have been positive, but even more so. Like I would think that the delta PFS would be even more. We can see that actually in the forest plot for PFS in this trial, that the greatest PFS benefit was in the Mayo 2018 high risk group. Now, they did not really give us the p-value for heterogeneity, which I would have liked to see, but there was clearly a trend from low to intermediate to high. There was a stepwise reduction in the hazard ratio in those three groups. So if at all, some of us will use it in the clinical practice down the line, we would only use it in the high risk group, definitely not in the lower intermediate risk group, because there is a concern for over treatment there. And one more point I wanted to make was that in the high risk group, despite that really good hazard ratio for PFS, I think it was 0.36 if I remember correctly, unfortunately, the PFS2 and the OS did not really show any signal in the Mayo 2018 high risk. Now, granted, it's a subgroup analysis, and the event number was low overall. So when you're looking at subgroups, the event numbers are even lower. But that kind of was a little bit surprising to me. I mean, given such a large delta in PFS1, I was expecting to see at least some kind of trend, at least in PFS2 or OS in the Mayo 2018 high risk. There was in the overall population, but not in the Mayo 2018 high risk.
So, Raj, as someone who is naive to the myeloma world, so they came up with this definition of high risk just by a group of people sitting in a room and saying that let's call this high risk smoldering myeloma and design clinical trial accordingly, or was it based on a large evidence?
So, basically, when they made this criteria, by that time, although there were two major risk stratification, like the Mayo 2008 and the Spanish, those were well established, I think, at that time. But there were also a lot of publications coming from like single center or multi-center studies, just looking at risk factors for progression of smoldering myeloma to myeloma. And one of the risk factors was, for example, immunoparesis. So if somebody has IgG smoldering myeloma, then immunoparesis of IgA and IgM, that's a risk factor. Those were not like formally in a risk score, but we knew from mostly the retrospective studies that those are also risk factors. So what they did in this trial was they included as many of the risk factors possible from the prior risk scores from these, individual studies, and came up with this criteria. I wonder, and I don't know the answer to this, I wonder if there was a discussion with the FDA at that time and FDA allowed, this criteria to enroll patients, because FDA is usually very, very stringent in when there is a biomarker that we are using to select patients. FDA is usually very stringent to make sure that the biomarker really aligns with the evidence. But I don't know whether that happened or not. And that was one of the concerns raised, that the inclusion criteria was a little bit too broad.
Probably another naive question. I know that the Mayo 2018 came out in 2018 and this trial started in 2017. Do you think they could have amended the protocol to include only patients with Mayo 2018 so that they are uniform in their definition? Because right now, I think, in my opinion, the challenge comes is we as a treating clinician seeing these patients in the clinic, how do we classify if they are high risk or low risk? Because a trial used a completely different definition. And right now, the patient in front of you is you are stratifying them based on the Mayo classification, right? Or
Yes.
the IWG 2018 classification.
Yes.
So how would you stratify that patient? I think that's a big concern.
Yeah, right now we would definitely use the Mayo 2018 until we have the genomic prediction models, which are currently being worked on. We don't know when they will become mainstream. But yeah, you're right. I mean, it's possible that they might have been able to amend. I mean, the Mayo 2018 publication was in June of 2018, and this trial I saw started accruing towards the end of 2017. It's a fair point that it could have been amended to make this trial more contemporary.
Now, I think to move to what I think is an even bigger issue, is progression-free survival meaningful in smoldering myeloma at all and in this trial?
Yeah, so that's I think the was the biggest argument and kind of the most I would say animated conversations that happened during ODAC was back and forth about these PFS events. So just to give the audience a background, so historically the smoldering myeloma trials, the primary endpoint would be either overall survival or it would be PFS, where progression would be considered as progression to crab myeloma, one of the four end organ damage: hypercalcemia, renal insufficiency, anemia, or bone disease. Now, in 2014, the diagnostic criteria for myeloma was amended to include SLIM, which was biomarker defined myeloma, which is serum free light chain ratio over 100, more than one lesion on MRI, and bone marrow plasma cells of more than 60%. And this trial, the primary endpoint was progression to myeloma, which was SLIM crab, so including the SLIM and the crab criteria. Now, a couple of things is, first of all, we now know that the risk of progression in patients with serum free light chain ratio over 100 is much lower than was initially thought, especially in patients with the 24-hour urine M protein of less than 200 milligrams per deciliter. That is a subgroup where the risk of progression is as low as 13%, 1-3. Definitely nowhere close to 80 to 90% that was initially thought. So that's one point. And the
You're
second
talking 2-year progression or 2 year progression?
That's a 2-year progression. Yes. So that's number one. And the second is, the reason these biomarker criteria were defined for myeloma is to be able to treat myeloma early and to prevent end organ damage. So one can argue that whether it's even an appropriate endpoint for a smoldering myeloma trial that we are treating one biomarker to prevent like another biomarker. So that was definitely a concern. And among the crab events, of course, we have all the four crab events, but clinically, the two most morbid crab events that we think of are first is a painful bone disease, for example, a fracture or a painful lytic lesion, and second is renal insufficiency. A mild anemia, which is not transfusion dependent, is not considered to be a morbid crab event. So just to go back in this trial, as we had said before, that the PFS was better in the daratumumab arm, but the question is that what kind of PFS events were prevented or delayed? I wouldn't say prevented because we don't see a tail in the curve yet, but were delayed. So the FDA had a really nice table. I think the investigators also did a great job of presenting the data in the initial publication.
Yeah,
So
we should mention to everyone that all of the presentations are available to watch on YouTube, and all of the FDA slides and the industry slides are available to download. So if there are any points you want to clarify, the slides are very easily accessible. But yes, go on, Raj.
yeah, basically SLIM events were much more common than crab events. And among the SLIM events, the most common SLIM event was actually serum free light chain ratio over 100. So that was the event that was prevented most commonly. So about, 33 out of a total of 67 events in the daratumumab arm were serum free light chain ratio more than 100, and 33 out of 99 events in the active monitoring arm was serum free light chain ratio more than 100. Then there was a fair number of patients with MRI, which I think, although it's a biomarker defined SLIM criteria, but it's clinically meaningful because we are catching a focal lesion early and preventing a lytic lesion. And then there were some bone marrow plasma cells. Among the crab events, the most common crab event that was delayed was anemia. And FDA did provide a really nice granular data on the anemia. And the median drop in the hemoglobin at progression was only about 1.7 grams per deciliter, and none were transfusion dependent or had fatigue as an adverse event. So that tells you that many of these patients were likely asymptomatic and were picked up on a routine blood work. And among the 14 patients that progressed with bone disease, just 3 had symptoms, and the rest were all detected on routine imaging. And there were no fractures from what we know. So overall, one of the concerns that FDA has.
And no renal failure either in either arm.
Yeah, I forgot to mention that. So there was zero renal failure in either arm, which I think is very reassuring when I have the discussion with patients that if they are going on active surveillance, the chances that they are going to, present one day with a renal failure is very, very low. And it goes along with the real world data that we have. And there were only one case of hypercalcemia, only one case that was in the control arm. So overall, one of the concerns that FDA raised that we are giving a patient 3 years of daratumumab to prevent mostly asymptomatic slim crab events or events which are not really morbid. So was that really a clinically meaningful endpoint? And I think we can discuss that in the next point on the overall discussion. But one thing I wanted to mention because it fits in here is that this is mirrored by the fact that at progression, about 20% of patients in either arm, the clinicians did not start anti-myeloma treatment. So that tells you that in those 20% of patients, the clinicians probably thought that one of the events that the patient had, the slim crab, was not really grave enough to start anti-myeloma treatment. So that again, raises question on meaningfulness of the slim crab endpoint, and that was a concern raised by the FDA.
I mean, on that point, one of the things that we talked about, and actually we had a lovely chat, you and I, along with Samir, with Vincent Rajkumar at ASH, and he spent a lot of time answering our very many questions, which was very generous of him. But I think for us, one of the reassuring things from this trial was to see the control arm and how well the control arm did in terms of the lack of morbid progression events. So sort of a funny takeaway, but I do think that for me is a big highlight of this trial is to say, if you monitor people closely, the chance of them having something, missing something is really very low.
Yeah, yeah, definitely. I mean, that is kind of the discussion that I'm having in clinic since the AQUILA trial was published, and that's definitely very reassuring to people. And one point I would make here also is regarding the bone disease. Typically, nowadays the recommendation is to do either a whole body MRI or a PET-CT. Sometimes PET-CT can be hard to get insurance approved every 6 months, but whole body MRI we are doing routinely, for example, at our center every 6 months for the first 2 to 3 years in high-risk smoldering myeloma. And the question is whether that will even decrease the number of lytic bone disease that we will catch and maybe transfer those events into MRI lesions, that we can catch patients at MRI lesion. There is a prospective trial called Spotlight currently undergoing to test that hypothesis. But I think that's another important point.
So in terms of talking about the results of the entire cohort, the hazard ratio is 0.49, favoring daratumumab, where we are seeing that 51% reduction in progression in DARA arm versus control arm. But if you look at the absolute difference, which is 63% versus 41% or 42% in the control arm. So the absolute difference is about 22%, 21%, right?
Yes.
So if you want to talk to the patient, and if I treat one patient in about 5 patients, you are preventing the risk of progression, right?
Yeah.
Yeah. So that is problematic to a clinician, because you're not preventing progression in 4 of those patients, but in one of them you are preventing.
So you've got 2 out of 5 who wouldn't have progressed anyway.
Yeah, that is correct.
And 2 out of 5 who progress even though you gave them the DARA. And it's only 1 out of 5 who would progress if you didn't give them the DARA, but didn't progress if you got the DARA. And all of that is progress in quotation marks. So, I think that's a great point, Ashman.
Yeah. So I think that's where I have the problem with this trial, is that they're treating all high-risk patients as same. And we are clearly seeing some heterogeneity in the definition of high-risk patients. We know that DARA works in myeloma based on all the multiple myeloma clinical trials with combination. But I think the question should not be whether DARA works or not in this setting. I think the question should have been more, which high-risk patients would have been benefiting DARA? I think that's where the trial, in my opinion, failed, because it doesn't teach me which high-risk patient I need to give DARA. They could have clearly done a continuous risk prediction model to show that which high-risk patient benefited from data. Yes, definitely, compared to control arm, the high-risk patients and data works clearly. But which patient in front of me should I offer data? They could have done a continuous risk prediction model, which they have not done. They just stick with binary yes or no.
So I think that leads me to a good next question I want to ask Raj, which is, Ash, when you say DARA works in myeloma, I think there's relatively wide consensus that that's the case. So what proportion of patients in the control arm that we're getting active observation here, Raj, actually received daratumumab at progression?
Yeah, so that was the third point is regarding the PFS2 and OS, because again, the investigators and the sponsor did a great job in presenting the data of PFS2 and OS. Especially PFS2 is not presented in all myeloma trials. So that was good to see. So now when we look at the PFS2 and OS results, one of the important questions comes is what percentage of control arm got DARA as in frontline myeloma treatment, and only about 1/4 of patients in the control arm, around I think 23 or 24%, if I remember correctly, received DARA-based frontline regimens at myeloma diagnosis, which is the current standard of care in US, UK, and Europe. So I think that was one of the problematic things when you are especially interpreting the PFS2 and OS results, because you might be looking at DARA now versus DARA never, not a question of DARA now versus DARA later at myeloma diagnosis. And as I had mentioned before, 20% of patients progressed to myeloma did not initiate any treatment. And also, in those who initiated treatment, the time to initiate treatment was very variable. There was a big, ranging from 1 month to I think around 49 months. So clearly, for some patients, even though they initiated treatment, the clinicians chose to initiate treatment much later. And one other thing that I wanted to really see, which I don't think we have the data on that yet, is in patients died in the control arm, how many of those patients saw DARA at any time point before death? So not only first line, but like second line or third line. And I don't think we have the data on that. Or even in the overall population, like what percentage of patients got DARA at any line of treatment after myeloma diagnosis, like second line, third line. They only give the first line data. Because, a substantial proportion of patients may have been enrolled in countries where DARA may not have been available. Are we just looking at the impact of early DARA, versus DARA never? That remains a question which makes it very difficult for me to interpret the PFS2 and OS results, which both had a trend in favor of DARA. To be fair, overall with the results, both had a trend in favor of DARA. Still, they did not meet the criteria for statistical significance yet, but you can clearly see that the hazard ratio was in favor of DARA, and the upper bound of confidence interval was just short of 1. So it was a wide confidence interval because very few events so far, but definitely both were in favor of DARA. But because of these problems, the post-protocol treatment, the overall survival results are very, very difficult for me to interpret.
And what did you make of the information they presented about causes of death in both arms?
Yeah, that's, very small numbers of deaths overall, so it's hard to know. First of all, FDA pointed out that there was limited information on the causes of death following subsequent treatments, whether it was from adverse effect of subsequent treatment or things like that. Death from progressive disease there were 2 deaths in the daratumumab arm, and there were 5 deaths in the active control arm. So numerically, slightly higher in the active control arm, but again, overall, 2 out of 194 in the DARA arm and 5 out of 196 in active control arm. And regarding deaths following subsequent therapy, again, we have limited causes of death, so it's hard to make any definitive conclusion surrounding that. And FDA discussion highlighted that uncertainty around interpretation of the overall survival signal here.
So, Raj, do you also want to talk about the grade 3, grade 4 adverse events that were reported in the DARA versus the control arm?
Yeah.
Because once again, I know that comes to the point. I think this is an important point to discuss because when you are discussing this treatment option to an asymptomatic patient, I think along with benefit, we also need to talk about the risk involved with this treatment.
Yeah, so actually, I did not even so far think about toxicity. The reason being that overall, I must say that daratumumab or isatuximab and CD38 antibodies are some of the best tolerated drugs in myeloma. I can't think of actually any other myeloma drug that I use in clinic which are better tolerated than anti-CD38 monoclonal antibodies. So overall, it's very well tolerated. So toxicity was actually not one of my biggest concerns, if I'm convinced about the efficacy here. Overall, to answer your point though, the risk of grade 3 or higher infections was about 16% in the data arm and 4% in the control arm. So it was roughly about 4 times. They did not report any infection-related deaths in the daratumumab arm. And yes, there were grade 3 or higher toxicities in terms of infection, but we see that when we use data in the real world also, but most of them are fairly manageable. So toxicity actually was not one of my biggest concern, even though that was a concern raised by the FDA. But I think if we think that this drug is really helpful, that will not stop any of us from using it.
I would add time toxicity though as an important point for people who otherwise can really have a blood test once every 3 months and maybe a scan every 6 months, or maybe blood tests every 2 months and a scan every 6 months, and see their doctor once every 3 months. They're now having to come every month for an infusion.
Frequently every week in the first 2 months and every other week in the next 4 months. Yeah.
Okay, so quite frequently. And then I think for pretty much every health system that's not the US, the financial side of things becomes very important too. Is this worthwhile? We won't get into that, but is this worthwhile for a society to invest in?
Yeah.
What do you think, to sort of move to summing up, Raj, what do you think the FDA is going to do here? And what do you think clinicians will do if the FDA approves it?
Yeah, it's a very tough question. I don't know. On one hand, I have concerns about overtreatment if it's approved. In my practice, I'm observing most of my patients, but specifically for low and intermediate risk patients who got into this trial, if FDA approves it for the entire protocol-defined indication, I have concerns about overtreatment. On the other hand, I feel like that there are some patients in whom I have been, it's a minority of my patients, but there are some patients in whom I have been using daratumumab since AQUILA was approved. After a thorough discussion of the evidence with them, they are willing to take the chance to get 3 years of data to delay progression because they may have a life event coming up in the next 2 years and they don't want to get on like a full-blown myeloma treatment, which would be dara VRD followed by transplant. I can see in a patient who is older and frail, more than 80, in those patients, maybe if you give 3 years of data, maybe some of them you will delay myeloma progression forever. So there are these patients here and there for whom it might be beneficial, and an FDA approval will help us in at least giving it to the appropriate patients. So it's a very difficult decision. I don't know what FDA will do. They might approve it based on the ODAC was a 6-2 vote in favor of daratumumab. So they might approve it. The question is that how will their approval, the indication look like? Will it be for the entire protocol defined indication, or will it be only for Mayo 2018 high risk, which was again subgroup analysis. So I don't know how FDA will reckon with that. All right, so before we go to the STARGLO, I just wanted to pick Ashwin's brain since he's an expert in precursor disease in the space of myeloid malignancies as to how the myeloid world is handling this regarding interventional trials in high-risk precursor states like CHIP or CCUS. Ashwin, can you tell us about maybe one trial that's ongoing in your field? How the inclusion criteria looks like, how are the endpoints looking like, and how is the lay of the overall field?
Yeah, I think similar to this trial, we in the myeloid world also are facing this challenge of what is called a high-risk CHIP or CCUS. Right now, most of the clinical trials which are being run in a phase 1 and phase 2 setting, we are not there in the phase 3 setting yet. But in the phase 1, phase 2, I think that the challenge we are facing is what is called a high-risk CCUS. So that is completely dependent on the individual clinical trial. There is no uniform definition. There is a study by Weeks et al. Published in New England Journal of Medicine defined high-risk seekers, but that was completely based on a UK-based healthy cohort population that may not directly apply to the patient in front of you in the clinic. So that's a different set of patient population. So there are other risk models being developed right now, but none withhold the testament of time in terms of validating in a different cohort. So it was all in one single cohort rather than there's a validation set. So that's the biggest challenge right now. So that is where I think, at least in myeloma world, I think we probably are having a better definition of what is a high risk patient versus a low risk, but we do not have the same definition in myeloid world. But in talking about the endpoints, I think the biggest endpoint in all these clinical trials we are implementing widely is improvement in cytopenias. That has been the biggest endpoint where the definitions are very clear. So that endpoints, I think, in my opinion, is clinically meaningful for the patient. And the other endpoint, which is secondary, is preventing the progression to myeloid neoplasms. But then the bigger question that comes is, what is the definition of a myeloid neoplasm? Because it's evolving there was a recent presentation at ASH 2024 which showed that the survival of high risk CCUS patient is very similar to low risk MDS. There is no difference, and which was thought provoking that these patients have similar survival. Then how can we draw the line between, without any intervention, we are seeing the similar survival? Then how can we draw the line? So those are some of the challenges we are facing in CHIP and CCUS, and I can talk about it all day. But I think definitely, I think myeloma world is much more advanced in terms of having a clear definition what is called a high risk, and have a phase 3 clinical trial already published. But we are not there yet.
All right.
Okay, so let's move to talk about STARGLO. Time for some lymphoma. If you, of course, haven't heard our episode after last year's ASCO and EHA, which we did with Dr. Gloria Iacobone from Barcelona, we discussed STARGLO in a bit of detail then, so highly recommend listening to that episode. But we'll talk a bit more about it now and about the ODAC that ensued just after, or just before the AQUILA ODAC.
All right, so to give a brief recap, MAB is a bispecific antibody targeting CD3 on T cells and CD20 on the malignant and benign B cells, and it's currently approved for use as a single agent in a third line or later DLBCL. So in its phase 2 trial, in which about a third of patients had relapsed disease following CD19-directed CAR T cell therapy, the complete response rate was 39% and the overall response rate was 52%. And the 1-year PFS was roughly about 37%. So logically, given its activity in multiply relapsed DLBCL, logical next steps were to move it to earlier lines of treatment. Now, of course, the standard of care for second-line DLBCL is CAR T cells for patients whose disease relapses within 12 months of frontline therapy, or chemotherapy salvage followed by autologous transplant in those with late relapses. However, prior to CAR T cells, there were a very limited chance of cure for patients who are unfit for autologous transplant, probably about less than 10%. So the STARGLO trial is a phase 3 randomized controlled trial, open label, in 274 transplant ineligible patients with relapsed refractory DLBCL. That is in the second line, which compared glofitamab plus GemOx, gemcitabine and oxaliplatin, against rituximab plus GemOx. On first glance, the results of STARGLO look unquestionably positive. The primary endpoint, overall survival, was significantly better with glofit GemOx arm compared to the R-GemOx arm, with a hazard ratio of 0.62. And after a median follow-up of about 21 months, the median overall survival was 25.5 months in glofit GemOx arm compared to 12.9 months in the control arm, so roughly about double. And furthermore, the median centrally assessed PFS was 13.8 months versus 3.6 months. And the centrally assessed complete response rate was 58.5% in the glofit GemOx arm compared to 25.3% in the control arm. There were a couple of extra infection-related deaths, so about 12 versus 4 grade 5 AEs in the glofit arm versus the rituximab arm, which was consistent with prior literature, but this was not enough to affect the overall survival results. There was an overall survival benefit despite this infection-related death signal.
So, Eddie, who can argue with this overall survival benefit in a randomized clinical trial? And the sponsor called it indisputable in the FDA ODAC.
I think when we saw the trial published, few people would have expected us to end up where we are now, but on closer inspection of the data, the STARGLO trial becomes a little bit more intriguing. So of the 274 patients that were enrolled and randomized, 88 were from Europe, 25 from the US, 160 were predominantly from Asia, and a few from Australia. So 52% of the intention to treat population, just over half, were from Asia, and most of those were from China. Now, this is where it gets interesting because the sponsors pre-specified region and race as subgroups of interest and pre-specified subgroup analyses. And patients from Asia, whether assessed by region or by race, there was a lot of overlap between Asian region and Asian race, which is not surprising. Patients from Asia in the GlowFit GEMOX arm had much better outcomes than those in Europe and North America, and Asian patients in the control arm had much worse outcomes than those in Europe and the US. And so the majority of the trial's positive result was driven by the results in the patients from Asia. I should say that the GlowFit GEMOX were a little bit different, but the R-GEMOX, the control arms, were really very different. So, for example, the hazard ratio for overall survival in Asia was 0.39 favoring GlowFit, while it was 1.06, so no benefit in the non-Asian patients. And really interestingly, you might think, oh, well, maybe that's just because they got CAR-T when their disease progressed. But actually, the same trend was seen in overall response rate. The difference in overall response rate was 46% in Asian patients, 9% in non-Asian patients. The same in complete response rate, the same in progression-free survival, as well as the overall survival I mentioned. So all of the efficacy endpoints somehow saw a much bigger difference in Asia than in non-Asian patients.
So, Eddie, like as a naive question, is the trial was powered to look at these individual differences in races?
So, this is all sort of a controversial area. You power the trial for the primary endpoint. You can't power the trial for a subgroup analysis most of the time, because then you end up with a way too big trial. But and it gets also messy because sometimes the sponsor would define Australia as being part of Asia Pacific on some slides in the ODAC, and in other slides they put Australia with the rest of the world. The FDA was pretty consistent that Australia belonged with the US and Europe, but the sponsor changed it a bit. I think this sort of gets to the key question of this whole ODAC, which was not so much about the drug as the trial itself. And can we really trust these trial results if there's such a big difference in these two populations?
One question I had was, can you think of any biological rationale or any reason why the control arm would do worse in Asia and mostly in China, and why even the response rate would be lower? As you said, the overall survival, we can come up with many hypotheses, but why even the response rate would be lower? Is there any difference in the frontline treatment in these countries? Doesn't look like, right? It's pretty much the same in all across the world, right?
Well, so yes, we're lucky in DLBCL that frontline treatment is R-CHOP pretty much, and as certainly until the last couple of years, definitely R-CHOP by 6, and that's been the case for sort of 20 years. But so look, I think the answer is we don't know. So the FDA proposed some hypotheses. For example, was a slightly higher rate of primary refractory disease in Asia, 59 versus 53%. There was slightly higher rate of relapse within 12 months, which includes the previous group, so 81%, 64%. So you have more patients in the non-Asian group who've had a late relapse, and late relapses typically do well and are more chemosensitive. And then cell of origin, which again was disputed between the sponsor and the FDA, but there was a substantial difference. So 70% of Asian patients had ABC cell of origin, just 23% had GCB, whereas 42% had ABC and 45% had GCB in non-Asian. So it's very hard to know how much that factored in. But while I'm on that topic, there were a few other areas that the FDA looked at to try and explain the difference. Another one was age. So median age in the Asian patients, 62, non-Asian 71, so a 9-year gap in median age. And very interestingly, 55% of patients were under 65 in Asia, whereas only 21% were under 65 in the non-Asian patients. And that is really important because this is a non-transplant eligible trial. And so if you're under 65, you really want to have some really good reasons to not be transplant eligible. But it's even more important in the CAR T-cell era, where we know from ZUMA-7 that there's an overall survival benefit to second-line CAR T-cell therapy. Glofitamab hasn't been tested against CAR T-cell therapy. And so, I think it gets really tricky when you have more than half of the Asian patients are under 65 in what's meant to be a transplant ineligible population. And that gets even more tricky when you look at the reasons given why the patients were transplant ineligible. Again, these data were disputed between the data the sponsor gave and the data the FDA gave. But in the FDA's data, 65% of patients from Asia were transplant ineligible because the patient refused the transplant. Only 7% of patients non-Asia were ineligible for that reason. So it brings out all this conjecture of, if you've got these younger patients who are ineligible because they refuse transplant, is that because they're somewhere that they can't get access to better therapy? Is it because they're being, doctors are saying, oh, I've got this great trial, yes, it's meant to be transplant eligible, but we don't, so this is all speculation. But because the differences are so stark, 65% versus 7%, a 9-year difference in median age, it raises questions.
Yeah, I think that is a strong point that the FDA mentioned. I mean, I can clearly see that maybe in Asia, what happened is that there were more patients who were early relapsers after R-CHOP who would really have been good transplant candidates, but they were enrolled in this trial instead. And, at the end of the day, those early relapses, we know that they don't do well on chemo alone. So, glofitamab is definitely a more powerful drug. And that's where, the other question I had was that, yes, there was a difference in the infection risk. Was that also driven, like, for example, were most of the COVID deaths in non-Asia or, versus Asia? Or do you know that information or no?
I don't think they gave the breakdown of infections specifically by region. I think they just gave the infections by arm. I mean, I think your previous point really gets to it, though, which is that so many of these characteristics are hard to measure. You and I can be at the end of the bed with the patient and see two very different risk patients, but these are all things that the FDA has identified that make you wonder if it's a different type of patient getting into the trial in one part of the world to the other part of the world. And I think, you said glofitamab is a powerful drug. I think it is, and I think that there's relative kind of consensus among lymphoma doctors that these bispecifics, glofitamab, epcoritamab, are powerful drugs. And I saw a number of people, particularly on social media, saying that this was a negative vote against glofitamab. And I think it's really important to highlight that the FDA said very clearly, the FDA is not seeking the advice of the committee as to whether STARGLO should be used to convert glofitamab from accelerated to traditional approval. They are specifically asking the committee to discuss if the results of STARGLO are applicable to a US patient population. So the FDA framed the question very specifically, very, quite narrowly, and I think very cleverly, to say, actually, the reason we're discussing this trial is not just about this trial, it's about a wider point we want to make to the kind of drug companies and to trialists, that you need to have enough US patients and enough consistency between patients from different parts of the world for us to be sure that the population, even if the population is not majority American, they made the point that the average number of US participants in global clinical trials is only 20%. So, whereas this was 9%. But really what they were saying is, you need a population and a trial that is representative so that we can apply it to US practice. And one element of that is recruiting enough patients in the US. but another element of that is design, control arm, evaluation. All these things have to match up with US standard of care. And I think that the vote and the discussion with the committee was much more around that very specific question of does it apply to the US rather than is glofitamab a good drug?
I think, Eddie, you bring up a very, very good point, and we see this all the time in the phase 3 randomized clinical trials in myeloid world as well. One trial I could think of is the luspatercept randomized clinical trial in the upfront setting, where if you look at the trial breakdown, only 10% of patients were US population and 90% were European and Asian patient population. And the company, FDA has the same question: is it applicable to US population, given only 10% were represented in the clinical trial? But I think the company made the argument that it's not FDA approved or EU approved or approved in other countries apart from US, and a lot of physicians were using in the frontline setting without having an approval in US. So that's why they had a lot of challenges in accruing. Is there a similar kind of explanation why we have seen a very less US accrual in this clinical trial?
Yeah, so the sponsor put a nice figure up which shows the recruitment by region over time. And really, I think there are two reasons. One is that I think it takes US sites longer to get set up and approved and ethics and all that sort of stuff. And I think that's still an issue. And second, they blamed a lot of it on COVID. And I sort of half by that reasoning, because I think the waves of COVID were earlier in China, but lockdowns were pretty prolonged and pretty severe in China too. So I think that is interesting. So another really important point was that the FDA went very hard about the fact that there was in the protocol a minimum number of Chinese patients to be recruited specified of 80. And they said that if they didn't recruit 80 Chinese patients in the main population, they do an Asian extension cohort, which is what they did with Polarix. There's an extra a couple of hundred patients that were added to Polarix specifically to get regulatory approval in China. And so I think part of it is the FDA saying, hang on, if you're going to put a minimum number of Chinese patients in the trial, where's our minimum number of American or American applicable patients? And I think that was sort of really a big part of this ODAC, was to say to everyone who was watching, you need, so we've had STARGLO, which is the frontline glofitamab trial, open at the center I work at, and it's now been closed for many months to recruitment in Australia, but it's still open, as far as I know, to recruitment in the US many months later. So certainly Genentech has learned their lesson from STARGLO, but I think COVID only sort of half bought that explanation. I think there is still a lag in terms of infrastructure between how quickly some of these trials can get going in the US compared to how they can get going outside of the US. And I think that would be a really good take home from a positive to come from this ODAC, would be to see that some of the regulators working on trying to reduce the amount of bureaucracy, paperwork, all that sort of stuff that does result in some of these delays. There are a few little things I want to point out just for anyone who's watching the ODAC, some observations that kind of irk me in terms of the way that the sponsor handles data. So if you look in the sponsor slides, they talk about there being 93% concordance between investigator-assessed progression-free survival and centrally assessed progression-free survival. This was a big issue in the CodeBreak trial of sotorasib in lung cancer, where they had people taking their patients off the trial earlier than the central assessment. And the problem here is that they don't give this concordance by region or by trial arm. They just gave us the overall number to say, look, it looks great, but I want to know what's the concordance in each of the four, the control arm, treatment arm, intervention arm, control arm, intervention arm in Asia and non-Asia, to see, is there a difference there? They also talked a lot about new anti-lymphoma therapy, or NALT. And we would often call that post-progression care. So there was a lot of squabbling over well, if you have a good response but not a complete response, then you might give a second, you might change treatment to because in DLBCL, what you really want is a complete response because you're aiming for cure. And so the sponsor made the argument that not only was overall survival impacted by NALT, next lymphoma therapy, but also progression-free survival. But then they put a slide up showing sort of the timeline of when patients got a second and next lymphoma therapy and NALT in the overall survival, but they didn't show that. They made the argument that it affected progression-free survival, but they didn't actually show that data. And they kept making this argument, but then they could have, if they wanted to make that argument with the data, they could have. So I think it's really important when you're watching sponsor-presented data that you think very carefully about what they're choosing to put on the slide and what's not presented as well. So, and on that same point, they presented event-free survival, or EFS, in the non-Asian population, but they didn't present EFS in the Asian population. So, it's really important when you look at each of these things, you think, hang on, where's the full picture? So I think I just wanted to highlight those couple of little examples.
Yeah, no, thanks, and Eddie. And then also, one more point I wanted to make is I think it would be a great precedent if, maybe going forward, FDA kind of forces companies to put in their protocol a certain percentage of patients that they have to accrue in the US, because I think it also has huge implications for overall survival and PFS2 results, because then, patients in the US will presumably get a contemporary standard of care at subsequent time points. And typically, as we all know, most drugs are usually approved first in the US. So I think that will really help in making the overall survival and the PFS2 results also contemporary. And, I think it will be a good thing overall.
Yeah, absolutely. I think that would be a great outcome. So to sum up, I think there are two important conclusions here. The first is that I think glofitamab is effective in DLBCL, and I think glofitamab GemOx is a great option to have because there are many patients who can't wait for CAR T cells, who don't want to move somewhere that they can get CAR T cells, who are genuinely unfit for stem cell transplant. And, we don't know about this population, but certainly patients with post-CAR T relapse might benefit from this regimen too. And I think it's important to think about, I guess that's my feeling. And I think we need more data to really reassure us of that, because the second point, which is that really what this data says about STARGLO is it makes you wonder about how much you can rely on these data. Even as someone who thinks this is a really good drug that I will use, a lot in my career treating lymphoma patients, it makes me worry about this trial because there's such a huge variety and so many different factors between the two halves of the population in the trial. And so I think it raises a sort of uncertainty about the internal validity, the robustness of the trial. And I think ultimately the reason why it was a kind of unanimous vote, not against STARGLO, but against whether it can be applied in the US, is really because of uncertainty. Is this due to biology? Is this due to race? I don't think so, but I can't be sure of that. Is it due to practice patterns? One thing I didn't mention is that the patients in Asia getting R-GemOx had their scans earlier than the patients getting R-GemOx outside of Asia. And, is that because the patients had clinical progression, so they had a scan earlier? Or is it because practice patterns were different? Something else came along, patients were disappointed they were in R-GemOx, so they had an earlier scan. We don't know. And I think that ultimately the vote comes back to that it's a question of not that this regimen, that people don't think this regimen works. I think there's genuinely still, fairly widespread consensus that it is an effective regimen, but just that this trial itself, it doesn't have sufficient certainty because of all these different factors we've mentioned. So that was our first emergency podcast, our first rapid fire podcast with just the three of us. And we'd certainly love to hear what you think. So feel free to reach out to Raj or Ashwin or me if you have thoughts or feedback. And thanks for joining us. And we'll be back with a widely acclaimed expert for our next episode. So get very excited about that. And we'll see you then, or indeed at EHA or ICML. Please come up and say hi if you see us there.